The overall goal of this procedure is to probe the composition of protein complexes that specifically assemble around a pair of interacting proteins of interest using Split-BioID, a protein fragment complementation assay that induces proximity-dependent protein biotinylation within living cells.
This method can help answer key questions in cell biology, such as how protein complexes dynamically remodel to regulate different cellular functions. The main advantage of this technique is that it allows the easy identification of context-specific protein complexes in their native cellular environments and with very high resolution.
For the final mass spectrometric analysis, the following steps are to be performed in keratin-free conditions, and all material and reagents should be as keratin-free as possible. After cloning the ORFs of 2 proteins of interest into the Split-BioID plasmid, carrying out transient transfection and adding biotin supplemented medium according to the text protocol, use PBS to wash the cells twice. Add 1.5 milliliters of PBS to each plate and use a scraper to harvest the cells. Then, transfer the cells for each condition to a separate 15 milliliter tube, and spin the tubes at 1200 times gravity and 4 degrees Celsius for 5 minutes. Remove the supernatants and snap freeze the pellets in liquid nitrogen. Then, store the tubes at negative 80 degrees Celsius until further processing. To prepare cell lysates, use 1 milliliter of RT lysis buffer to resuspend the pellets. Then, pass the cells through a 25 gauge needle.